when Judith and Lucy and I first decided that we were going to go ahead with this idea, we were looking for a good communicator, and we were looking for an interesting talk. And when we narrowed it down slightly, I mean, you know yourself, you do a little brainstorming session. Within about 2 minutes, we came up with the idea that we wanted someone to talk on genetic deformities in household pets. Preferably with a link to mid-20th century fiction, if possible. So we had a little look through the many speakers that satisfy that criteria. And we settled on Kat, and we decided that even though Hemingway's work was mostly in the first half of the 20th century, we'd let you away with that. No. What we really wanted was someone who was going to come, be passionate about what they talk about, be interesting about what they talk about, be able to communicate their passion to non-biologists, to non-scientists, even though the idea of genetics is something that defines, in part, who we are, what we look like, the way we act. So, talking on herding Hemingway's cats, how genetics affect our lives, Kat Arney.
Thank you very much for having me. It's really fantastic that so many people have turned out, and it's just really great to support community events like this. So it's a real privilege to be here. We hear about genes all the time. They're the things that they make our eyes blue, they make our hair curly, they make us fat, they give us cancer. But, like, how do they work? Right? So I'd say this to my friends, and my friends would be like, I don't know. And then I went to talk to all the scientists I knew. So my background is in genetics. I went to talk to all these people that I knew who were working in things like developmental genetics, developmental biology, searching through the genomes of all sorts of people and organisms. And I said, I want to write a book about how genes work. And they all said to me, well, when you find out, let me know. And that really made me think that not only was this going to be an incredibly challenging book to write, covering the whole of genetics, but also that it's a very exciting time to be trying to figure out what we know about how our genes work and what's in our genomes. We're just getting to the point where we can read an entire person's DNA sequence, their genome, for under £1,000, $1,000 or less. We're finding out all these things that are within us, how our genes interact with the environment to make us who we are. But we still don't really understand how it all works. So I went around the world, I talked to a whole bunch of scientists. All the book is written as first-person interviews with these people, explaining some of the stories that I've encountered along the way. And so I thought I'd do a quick talk about some of the things that are in the book, talk briefly about the Hemingway Cats and what I discovered about them, and give you a bit of an introduction to some of the things we do know and what we don't know about how our genes work. Two of the things that I really love are the internet and science. And it turns out that one thing that links these two things together is cats. Always. Lots and lots of science-based jokes about cats, cat-based jokes about science, but it turns out you can't make cat-based jokes about science on the internet too often. So, also it turns out that DNA is made of cats. Cats are also made of DNA. Or, to put it more sensibly, this is DNA. Many people familiar with the idea that this is the structure of DNA, the double helix that was discovered by Watson and Crick and Franklin and Wilkins back in the 50s. It's the blueprint of life, the architect's plan that makes us who we are. It's within all our cells. It tells our cells what to do, when to grow, when to multiply, what to be, when to die. It's important stuff. It's also a very handy, lazy advertising shorthand for like, woo, science. But this is the molecule of life, DNA. I'm going to talk a bit more about it, but just know that it's a double helix, this twisted ladder, that's made of pairs of chemicals all stuck together in a row. So, DNA contains genes. Genes are basically recipes. So small stretches of DNA are genes, and this is pretty much like the idea that all your DNA, your genome, that's the sum total of the DNA you have, is like a massive library of recipe books, and each recipe is a gene. And genes make things in cells. There's no gene for Alzheimer's. or gene for schizophrenia. There's no gene that makes your big toe or makes your arm. Genes are the recipes that tell our cells to make molecules called proteins. Sometimes they tell our cells to make molecules called RNA as well, and that's how we make all sorts of the molecules in our bodies. But the key thing to know is genes are effectively recipes. And so when people say, oh, the gene for schizophrenia, no, it doesn't exist. What they'll have found is, usually in the cases of when you read the headlines that say 100 genes for schizophrenia found, what they'll have found is 100 places in the DNA that seem to be a particular way in people who have schizophrenia. What they haven't necessarily found is a molecule that gives you schizophrenia. So that's what they haven't found. Genes are recipes that tell our cells what to do. And so DNA is the recipe book of life, and we need to know a few things here. So is anyone here a librarian? A librarian, excellent. I love librarians. My mother is a librarian, an archivist. But you guys are kind of picky, aren't you? Oh yeah. You like the books to be kept clean and neat and tidy, and you like things all to be in order. And say if I wanted to come to your library and get the latest Mary Berry recipe book and cook a cake from it, you would be cross if I turned up with my mixing bowl and all my flour and everything in your library and started cooking right there, wouldn't you? And cells are very similar. So we keep our DNA, our recipe books, nice and safe and clean and sound in a structure called the nucleus, which is in the middle of all our cells. And this is basically, DNA is important, it's your instructions. So we keep it in the middle of the cells, nice and safe. And all the cookery, all the mess that goes on, the making the proteins and all this kind of biochemical stuff, goes on outside the nucleus in the cytoplasm, the kitchen of the cell. And this is how it works. I apologise in advance for my really terrible PowerPoint animation here. So, we have a piece of DNA, that's DNA. A stretch within the DNA, a gene, effectively a recipe. And we want to make this recipe. We want to make the protein that this gene has the instructions for. So what happens is, just in the same way that you can't, this is a reference library, you can't take the books away, but you can photocopy them. So we're going to make a photocopy of this gene that we can then take away into the kitchen, like this. Ta-da! It's a molecule called RNA polymerase, and it makes a copy of the gene from a molecule called RNA. It's very similar to DNA, it's effectively a copy. So we've made a copy, and it goes out into the cytoplasm, into the molecular kitchen, where all the mess and the chaos is. And then there's a molecular chef called a ribosome that reads it and makes the protein. And the protein then gets kind of folded up and does, there we go, does what it needs to do. So what are these proteins? You might hear about proteins in the context of things like, meat or eggs, that's meaty stuff. But our proteins are the building blocks of our cells. You're made of a trillion cells, roughly, and you have skin cells and bone cells and brain cells and heart cells and liver cells, and they're all doing their own jobs because they make different proteins. So your skin cells are packed full of a really sturdy protein called keratin. Keeps the outside out, keeps the inside in. Your brain cells are fizzing with neuroreceptors and neurotransmitters, chemicals and proteins that send signals to enable you to listen to my talk. If you're digesting a big Sunday lunch, who had a big Sunday lunch? Was it good? Yeah. I had a bag of crisps in Reading Station. If you're digesting your food, the genes that make the enzymes and the hormones that help you digest your food and use it are making these proteins. So we're packed full of all these proteins, and the genes are the recipes that enable our cells to make them. Does that all kind of make sense? That is effectively the first year of a molecular biology degree. So you can pick up your certificate at the door. Well done. We've got that. So fundamentally, that is how your genes work. Now, we could just stop here and go, case closed, we know how genes work, photocopy and it makes it, blah, blah, blah. So here's the problem. So as I said, you've got skin cells and bone cells and brain cells, all these different kind of cells. But they all have the same DNA. They all have the same recipes, but they're not all making the same proteins. So clearly, things are a bit complicated here. And cells have to decide which genes to use at the right time, in the right place. Because the cells in your skin don't suddenly go, you know what, I'm bored of being skin, I'm going to go and be a brain cell. No, they stay doing what they're doing. So how does that work? Well, we'll come to that. But first I'll give you some of my favourite facts about DNA. And the next fact, this is amazing, my favourite fact. You have over 2 meters of DNA in every single cell of your body. This boggles my mind. 2 meters. It's very, very thin, I'll give it that. But 2 meters of DNA in every cell of your body. It's twisted and crushed and wrapped around kind of ball-shaped proteins and squeezed and coiled and coiled and coiled to fit into a structure smaller than the head of a pin in every cell of your body, or less. And that is enough DNA to go to the moon and back 1,500 times in your body. It's incredible. I always find it amazing when, Brian Cox, we love Brian Cox, and he looks up at the stars. He's like, the stars, they're amazing, billions and billions and billions. Like, look in your own body, Brian. It blows my mind. But everything I learn about biology just blows my mind. That's probably why I'm less famous than Brian Cox. So this 2 meters, it's not just like a piece of string that's 2 meters long, because that would be really difficult for cells to work with. It would get tangled up, it's very difficult to copy, to divide, to read, all these kind of things. So in our cells, this 2 meters of DNA is chopped up into 23 pairs of what we call chromosomes. And chromosomes, you hear that word, it's just basically a length of DNA strung through with genes. So we have, if you're genetically a female, you'll have 22 pairs of matching chromosomes and then 2 X chromosomes. If you're genetically a male, you have the 22 pairs of chromosomes and then an X and a Y. And so that's kind of a representation here. This is a chap. You can see there's an X and a Y there. And within this 23 pairs of chromosomes, this 2 meters of DNA, it's made of 3 billion, what we call letters, bases, or base pairs. And these are the chemicals that form the rungs of the ladder of DNA. I have had people, because we hear about these letters quite often here, A, C, T, and G. I've had someone say, can you actually see the letters when you look at the DNA? No. These are chemicals. But the letter thing comes from the names of the chemicals, adenine, cytosine, thymine, and guanine. But what you need to know is that these are pairs of chemicals, A with T, C with G, that make up the rungs of this ladder. And that encodes, that's the 4-letter alphabet that all the recipes of life are written in. And it's a huge amount of data, it'd be like 200,000 page phone books. Huge amount of letters. And within that are our genes. Anyone who's not a biologist want to hazard a guess at how many genes we have as humans? Is that 1,000? Anyone? Higher? No? An awful lot. So there was this whole thing where they started reading the human genome back in the 90s. We wanted to know how many genes it takes to make a human being. And we had some clues. We knew how many genes bacteria had. We knew how many genes yeast had. We knew that things like little worms and fruit flies had about, 20,000 genes. And so we thought, okay, how many times more awesome are humans than fruit flies, right? We're pretty awesome. So we must have 150,000 genes. That sounds about right, doesn't it? We're more complicated than worms. Turns out not so much. When they actually started looking through the genome and working out what's genes and what's not, it's about 20,000 genes. Ladies and gentlemen, we are sophisticated worms in terms of our genomes. But the important thing to know is that we make many, many, many more than 20,000 different proteins. We make hundreds of thousands of different proteins and other sorts of molecules as well. So rather then each gene being a very, very strict kind of Delia Smith recipe. You know, heat your oven to 217.5 degrees, use this size tin, do exactly this, otherwise the hell mouth will open and your cake will not rise. These are the much more kind of sort of sexy, go as you feel kind of Nigella type recipes. You know, maybe try oranges, try apples, what about lemons? Whisk your eggs, don't whisk your eggs. These are suggestions you can chop and change. Substitute chicken for cashew nuts if you're feeling a vegetarian. There are lots and lots of different ways that each recipe can be interpreted and used as it's needed. So we actually get a huge amount of bang for our biological buck by using our genes in many, many different ways. So there's some wonderful stories there. And then how much of this genome, this 2 meters of DNA, this 3 billion letters, is actually genes, this 20,000 genes? Again, it turns out, not so much. Less than 2% of the DNA in each one of your cells is honest to God actual protein coding genes. So what's the rest? Is it just junk DNA? Has anyone heard the phrase junk DNA? It really winds me up. Because this phrase junk DNA, it started to be used in the 60s. It was actually used in the 60s in Cambridge first and then became popular. And no real geneticists have ever thought that all the non-coding bit of the genome was junk. Because of course it's got to have other stuff in there. But because it became a word that everyone used in the media, suddenly when they find something in it that's not junk, everyone goes, oh my God, the junk DNA is not junk after all. Or they find a piece that is junk, and they're like, oh, it's all just junk. So it's very misleading to refer to all of this stuff as just junk, because it's a lot of different things. So, a significant proportion, maybe sort of 5 to 10%, we're not entirely sure, is the control switches. Like I mentioned earlier with the Hemingway Cats, the control switches that turn genes on and off at the right time in the right place. There's quite a lot of the genome that we know gets photocopied into RNA. We know it's kind of red, it's photocopied. We don't know what that RNA does. Some of it we think kind of maybe fine-tunes the activity of genes. Some of it might be important for other things. Some of it might just be there because life is messy and 2 meters of DNA stitched into this tiny space and things accidentally get fed through the photocopier. We don't know. Just as with life, a significant proportion of your DNA is very dull and repetitive. You know, our genome is packed full of these long dead viruses called transposons, what we sometimes call jumping genes. They're viruses that a very, very long time ago got into the genome, copied themselves some more, copied themselves some more. You get hundreds of copies of these repeated elements just stuck there. And we don't know if they do anything important. We know some of them get co-opted for useful things, some of them don't. They're just there. And we really need to challenge this idea that evolution is this perfect winnowing fire that's made our genome incredibly efficient. It hasn't. Our genome is full of just stuff. Has anyone got like the cupboard or the garage that's just kind of full of stuff? I've had to move house a lot in the past couple of years, and I have boxes of stuff that I just pick up and just move to the next place. I'll go through that some point. Probably when I retire. Or I'll just throw them all in the sea. But it takes time and effort to sort through stuff. And it takes time and effort for evolution to sort through the genome, get rid of stuff. And if it's not doing us any harm, it just stays there. So some of it is genuinely junk. Our genome was not designed, it's not efficient. It evolved. And the key thing to know about evolution is it just cobbles stuff together. And it doesn't care if you have a beautiful, perfect, efficient genome. It just cares that you have babies. So, and some of our DNA is probably what we could call garbage. It's maybe actively a bit harmful to us. There are transposons that we think might be hopping about. There's a lot of stuff that we don't know about what's in there. But it's not all junk, and it's not all functional. It's a mixture of stuff. So I want to talk very briefly about some control switches. There's quite a lot of stuff in the book about how we use these control switches to turn genes on and off at the right time in the right place. And this is how we address that problem that I raised earlier, that we have all sorts of different types of cells. We have skin cells and bone cells and liver cells and pancreas cells and everything. And they're all turning their genes on and off in different ways depending on what they need to do. And this goes right the way back to the very, very beginning of development. You start life as one single cell of fertilized egg when mummy and daddy love each other very much. And you become 2 cells, you become 4 cells, you become 8, 16, a ball of cells with a tiny little splodge of stem cells in the middle of it. And that is going to be you. And by switching on different genes, switching off other genes, making decisions, are we head, are we tail, are we inside, are we outside, are we gut or liver, are we heart or muscle? All these decisions are made that build you as a person and every other organism, and also enable you, once you're grown, to respond to your environment, to respond to digest your dinner or regulate your temperature. And now, I'm going to tell you a story about some of these control switches that centres around Ernest Hemingway. Now, I love Ernest Hemingway for 3 reasons. Firstly, as a writer, I love Ernest Hemingway. I love the way he writes. I love his style. The casual misogyny and violence, not so much, but no one's perfect. As a drunk, I love him. And also as a biologist. Now, the reason I love him as a biologist is because he liked some things. He liked fighting. Man loved fighting, loved writing about fighting, bullfighting, war. Man loved fighting. Fishing. Man loved to fish. Wrote about fishing, The Old Man and the Sea. Loved fishing. Mating. And all of these things coalesce in one organism, which is the irony of this entire thing being, I'm a dog person, and now people keep sending me pictures of their cats and buying me cat stuff. I actually don't like cats at all. But anyway, specifically with regard to Ernest Hemingway, is these cats. This is a polydactyl cat, and you can see that rather than having the neat kind of paws there, he's got this kind of weird extra thumb claws business. It's almost like he's got a thumb, or she, not sure. Just says polydactyl cat, not sure. Polydactyl gender neutral cat has these kind of weird thumb business going on. Now the legend goes that Ernest Hemingway was given a 6-toed cat, polydactyl cat, by an old sea captain. And these cats were very, very popular on ships in the great time of seafaring, and you still find polydactyl cats all up the eastern seaboard of the US. They're all genetically the same, so they all come from one cat that's been bred and selected for. You also get polydactyl cats in the UK. They have a different genetic mutation. There's 2. There's some in the Midlands and some in kind of Cardiganshire in Wales, and they've spread through the UK. So there are polydactyl cats in the UK. Please don't send me photos of yours. I have enough now. So the story goes that these cats were very, very popular on ships, and I can think of a few reasons why. The advantages of this marine feline polydactyly. So the myth goes maybe that they were better mouses. The reason you keep cats on ships is because you want to get rid of the mice and the rats, and if you've got bigger paws, bang, you're better at catching things. The other idea, perhaps, is that because you've got a semblance of a thumb, you actually maybe have a prehensile grip as a cat. So when the ship's tipping around, you can grab on a rope like and not get thrown overboard. And these 2 things would have very, very strong selective pressure in evolutionary terms. Because if you can catch prey better, you'll be fed. And also, if you don't go overboard, then you'll be maintained by the breeding pool and not lost to the pools of the briny deep. There's, I think, one even stronger selective pressure that has meant that these cats have persisted in the way they have. And that's because sailors are really superstitious. And what's more lucky than a cat with 5 toes, right? So all of these very strong evolutionary pressure, but it all boils down to, in molecular terms, a gene that I'm going to talk to you about. This is a bit of molecular biology. So what we're looking at here is a stretch of DNA, and the gene we're going to focus on, the little stretch of DNA that is the recipe here, is this. Now it says shh. It's not a librarian's gene. It actually stands for a gene called Sonic Hedgehog. And yes, it was named after the video game character. There's a gene in fruit flies that was discovered called hedgehog, because when it's faulty in fruit flies, it makes the embryos all kind of hunched up and spiky, a bit like hedgehog. And so when researchers started looking in mammals for these genes, they found 3 versions of hedgehog, and they called the first one Indian hedgehog, because of like there's hedgehogs in India. They called the second one desert hedgehogs, after desert hedgehogs. And the third one was named by the postdoctoral researcher that had a 9-year-old girl. And he was looking through her cartoon books and saw this blue hedgehog. He was like, cool, let's call it Sonic Hedgehog. So a couple of lawsuits later, and it stuck. And there's a lovely, lovely story about the boss of the lab, where it was discovered, actually was so thrilled that their new gene was so popular that McDonald's were making Happy Meals based on it. That's genuinely a true story. So, Sonic Hedgehog, it's not, as I said, genes don't make things, they make molecules in cells. And so it's not a thumb gene or a finger gene, or whatever cat fingers are called, cat digits. What it does is it helps cells to make decisions. Helps cells decide, are you going to be this or are you going to be that? Makes a kind of a decision molecule, let's call it that. And it's involved in lots of places during development. So the blue circles here, those are some control switches. There's a control switch is a little stretch of DNA that binds molecules that help turn the gene on. They attract the molecular photocopier and say, let's turn the gene on here in this tissue. So that says CNS, central nervous system. That switches on Sonic Hedgehog in certain cells in the brain, and during development helps them decide, are you going to be a brain cell or are you going to be a supporting cell in the brain? It's switched on in the epithelial linings. The lining of the gut helps cells decide what they're going to do in the gut. And the thing we're most interested in with regard to the cats is the green one there where it says limb bud. And that switches on Sonic Hedgehog in the developing limb. So what we're looking at here, this kind of blob, if you imagine a developing fetus or a developing mouse, this is a mouse in the womb, this is the paw, the developing paw of a mouse. So they, about halfway through development, a third of the way through, they have these little kind of paddles instead of hands, because their hands haven't grown yet, and they sort of grow out like that. So at this stage, Sonic Hedgehog is switched on in the sort of what's going to be the fleshy bit at the bottom of your hand, if you can imagine that. And it spreads up through the developing limb bud, and it basically helps cells decide, are we going to do finger or not finger? Finger, not finger, finger, not finger, finger, not finger. So that's kind of how it works normally, and we have it, mice have it, cats have it, helps you grow your digits. But in the Hemingway cats, and also in some polydactyl humans, this switch isn't working properly. It's not that it's broken, it's not that Sonic Hedgehog isn't turned on at all. It's just more like it doesn't quite know where to stop. It's not turned off at the right time, and you end up with sort of going, okay, let's do fingers, not fingers, fingers, not fingers, fingers, not fingers, fingers, not fingers, fingers, more fingers, more fingers. So you end up with extra digits over here. And that's what gives the Hemingway cats their thumbs. It's also what gives some types of polydactyl human their extra digits. Now, I got kind of thinking about this. And, you know, that you don't want to be messing with genes so much, because genes are important. They're the recipes for our cells. And as I've just shown you, Sonic Hedgehog is important in the brain and in the gut, as well as the limbs. You know, if you want to evolve new characteristics, you don't want to be messing around with it. And in terms of the cats, you do sort of wonder where this evolution is going to take them. Can they pick locks, read books on military strategy, and kill us as we sleep? And I, for one, welcome our feline overlords. But this really got me thinking and looking for examples about where in development this playing with these control switches had had impacts on organisms and had maybe shaped evolution and shaped species. And it turns out there was a lot. So these control switches are evolution's playground, because you don't want to be messing about with the structure of the genes. And we know we can look across a lot of different species, and the genes, the recipes themselves, are pretty much the same. You know, our protein coding genes are very similar to mice, very similar to chimps, very similar to pretty much most mammals. We have mammal genes. But we have a whole bunch of different control switches that turn our genes on and off in different ways to make us who we are, make chimps what they are, make mice what they are. So I'm going to give you an example from the fishy world. These are sticklebacks, two different species of sticklebacks. Top ones live in marine environments, in the sea. The bottom ones live in freshwater lakes. Now, just kind of put it as best as I can for children in the audience, when the sticklebacks in the sea feel like going on a romantic holiday, they like to go on romantic holidays to the lakes. Don't we all? Beautiful. So they go on their romantic holiday to the freshwater lakes and have a lovely time, buy some ice cream, take some photos, all that kind of stuff that they do. And then they go back to the sea. Now the problem was, about 10,000 years ago, there's a bunch of sticklebacks that were having a romantic holiday in the freshwater lakes, and the Ice Age happened. And they couldn't get back home again. So, well, we're stuck on holiday forever. Brilliant. So that's what scientists call a speciation event. You separate two populations so they can't breed with each other anymore, and they can start evolving separately. And that's what happened. So if you look really closely, these fish kind of look pretty similar, to be honest. They're not separated by that much time. 10,000 years isn't that long in evolutionary terms. But the top ones, if you look kind of this area here, they have what I like to term death spikes. This is their protective mechanism. This is how they protect themselves from predation in the sea. A lot of predators in the sea. I'm not sure how effective it is when you're a 3-inch long fish, but it must work because they have them. Freshwater fish don't have the same predators. No death spikes. See, gone, missing. Now, when we think about what we may think we know about evolution, evolution is slow and gradual, isn't it? It's tiny changes. Something gets a bit bigger or a bit smaller, a bit larger, a bit fatter, a bit thinner, a bit darker, a bit lighter. These very gradual changes across millennia. Not so much in the case of these fish. So a researcher called David Kingsley at Stanford University in California, because he's a scientist and that's what they do, took some of these fish, put them in an X-ray machine. And what he saw was boggling. So the top fish are the marine sticklebacks. And you can see they have this kind of pelvic structure with the death spikes sticking out from it. The bottom fish are the freshwater fish. Not only do they have no death spikes, they have no pelvis. This is like waking up and finding your bum is missing. It's not that, oh, it's just a bit smaller, or they just become tiny vestigial things. Gone, vanished, gone, with the bum as well, gone. These are fish with no bum. How did that happen? And when David started looking more closely at the DNA of these fish, he noticed something in the control switches. So this all centers on a gene called PITX1. The PIT bit stands for pituitary. You have a PITX1 gene. It's involved in making your pituitary gland and your legs. I keep saying hind legs when I do something. No, just the legs. Centaurs don't exist yet. I'm working on it. So the PITX1 gene in fish, it gets switched on in their mouth and their jaw. It's important for structures in the head end. But it's also important for directing the formation of the pelvis and the death spikes. And when he looked closely, he found that in the marine fish and in the lake-dwelling fish, they had the gene was absolutely fine in both of these species. And there was a control switch that turned it on in the mouth and the head, and that was absolutely fine in both species. But the control switch that turned it on to make a pelvis was broken, it was gone, it was faulty in the freshwater sticklebacks. So they didn't turn this gene on, and they just didn't start the part of development that says, let's build a pelvis and some death spikes. So just by playing with these switches, getting rid of one of these switches, you're completely getting rid of a massive structure in development. And amazingly, what you can do, you can actually take the piece of DNA that's the control switch, you can take the gene, the PITX1 gene, you can kind of stick them together, make a little bit of artificial DNA. You can inject that into an early stage embryo of one of these sticklebacks that lives in the lake, and it will start growing death spikes. Look at that. First one in 10,000 years to have them. So, you know, these fish, they have the equipment to do this, but they just don't turn the gene on at the right time, in the right place to do it. And I started looking for other examples where this had happened, where evolution was playing with these switches to give us different characteristics. And I found one in a gene that's called KIT-LG. Again, it's a gene that does lots of different things in development. It's involved in skin pigmentation, in hair, it's involved in making eggs and sperm cells, in blood cells, it does a lot of different things. You do not want to go messing about with this gene, but we can fiddle with the switches. And it turns out if you have one letter different in a control switch for this gene, just one letter different, so we're not talking about a complete thing being missing or broken, a very subtle change. It's the difference between being a blonde and a brunette. So it's not like the fish where something's missing and an entire structure is missing. We're getting to the point now where this is more like twisting up the dials. This is a pigmentation gene. You're making a bit more in brunettes, a bit less in blondes. And there are other variations that give us the kind of range of colourings that we have. Same gene again, different control switch, single letter change. Is the difference between white skin and West African skin. And you know, you can have two versions of one, two versions of the other, one version of each. You get a range of shades in between. This isn't a yes/no. This is like fiddling with the dials of variation. And as I was writing about this in the book, I was watching the TV, and it was the time of the Ferguson race riots in the US, and the country's just tearing itself apart over one letter of DNA in 3 billion letters of DNA. That, you know, I talk to biologists, to geneticists, and they're like, we're all mutants. We're all just variants of who we are. It doesn't make you better or worse. We're just all variations. And, you know, we are really stupid people sometimes when we don't see that the variations in our DNA are just that. They're not better, worse, yes, no. They're variations. So that's the philosophical bit. I do have some slightly not safe for work bit, which I will try to make as safe. Well, I wasn't expecting children. I had to do this talk in front of my dad, though. That was even more embarrassing. Okay, here's the most not safe for work image I have. So these are photographs of the genitals of different species, male genitals of different species. Now, if you were to go on a romantic holiday with a chimp, ladies, you would notice something. They wouldn't pay for dinner, for a start. And the reason for this is that chimps are, let's put it like that, they have more kind of a wham-bam, thank-you-ma'am approach to the whole business than human species, who at least do tend to buy you dinner first, in my experience. And as a result, there are the species that have this kind of attitude to romance. They tend to have what I can only describe as penis spikes. They have hard spikes that enable them to cling on during mating. Human males, I've been led to believe, don't. Just doing this in front of my dad, I was like, been married twice. Human males don't, but these species do. So what is that? Because we're all mammals, right? And I think you're probably guessing where this is going now. It comes down to a faulty control switch. And the gene in question is a gene called the androgen receptor. Again, it does lots and lots and lots of different jobs during development. It makes a receptor for testosterone, the male hormone. It's the hormone that makes men kind of big and butch and beardy and manly and put up shelves and any gender stereotype you want to insert there in males from humans, here we've got chimps, macaques, and mice. And he found there was one crucial difference. So whatever the purple one does, that's all fine, that's there. Whatever the green one does, that's all fine, gives the mice beards or whatever. But in humans, they were missing this crucial control switch. And what it does is it turns on the androgen receptor in the developing male genitals. And in these species, that means they grow spikes. In human males, it is missing. Do not grow spikes. And they proved that this was actually what had gone on, that this was crucial, because they took, again, that control switch, the DNA for that control switch, they took the DNA from the androgen receptor gene, put them together, and put them into male foreskin cells, human male foreskin cells. Because it turns out you can buy anything on the internet. And they discovered that, that these cells started growing these kind of bizarre structures, as if they were trying to do, grow the spikes. So that shows, chaps, you have it within you. And I think I speak for all the ladies here when I say I'm glad that you don't. I've been led to believe. So I think I will stop there and take some questions. Thank you very much for your attention.